Good afternoon. Am I audible?
Good afternoon,
Yes,
sir.
sir. Assalamu alaikum.
Walaikum salam. Thank you.
Assalamu alaikum, sir.
We have only 13 students still. 14. Okay. How are you?
Alhamdulillah,
Alhamdulillah,
sir.
sir.
Very good.
Alhamdulillah, sir.
I am trying to show you the slide. If it is visible, please tell me.
Yes, sir.
Sir,
It's visible.
it's visible.
Okay. So what can you see? Plant tissue culture, right?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Yes. Now, full screen, right?
Yes,
Yes,
sir.
sir.
Okay, thank you. I welcome to today's talk on plant tissue culture. Unfortunately, we are missing face-to-face interaction today, but I shall try to tell the story of tissue culture. I guess many of you know about plant tissue culture, uh, but I shall try from the scratch to the science up to its application as a biotechnology. Plant tissue culture are also known as culture in the glass or in vitro culture. That means you cultivate plant tissues in the laboratory. Why? To make uh, many copies of the plant. So, how can we define plant tissue culture? Plant tissue culture broadly refers to cultivation of plant cells, tissues, organs, and plantlets on artificial medium under aseptic and controlled environmental conditions. Here, some key points are any parts of the plant you can cultivate on artificial medium, that is, culture medium, contains nutrient for the cell growth. And another point is aseptic. Aseptic means you do not allow to contaminate with microorganism and others, that is free from the contamination, and obviously controlled environmental condition in the laboratory. Temperature, humidity, and other things are controlled. Plant tissue culture is as much as an art as a science. So it is one of the classical biotechnology uh, which has been used industrially worldwide uh, since long, but it is still an appealing uh, technology for using in modern genetic engineering or genome editing. So it is the art of growing experimental plants, selecting a suitable plant organ or tissue to initiate cultures, cleaning, sterilization, trimming. These are the steps. First, you select a suitable plant or organ or tissue, and then clean it, sterilize it, and trimming it to a suitable size and planting it on a culture medium for its growth. So, what are the basic principles of tissue culture? the basic principle of tissue culture is the totipotency. Totipotency means plants or plant parts have the potential to produce, new plant. Among the eukaryote, that is uh, multicellular organism, plants are champion. If you take a small piece of plant, you can make the complex whole plant. And this is why it is called totipotency. You cannot find in animals, human, this totipotency, especially for the vegetative tissues. But in case of human, after uh, fertilization, the embryo is like totipotent or omnipotent. It can grow, multiply, and finally develop as a complete human baby. So totipotency we can define as the ability of a cell to differentiate and develop into a whole plant when given the correct conditions. So this is very important. When given the correct conditions, nutrition, hormone, and other conditions must be congenial so that a single cell of plant can differentiate and develop into a whole plant. This is because every cell has the genetic potential of the parent plant. This is the real principle of totipotency. So totipotency, if we demonstrate it here, plant cell, a single cell, and then it can grow, multiply, and produce the callus. Callus means profusely multiplied cell, undifferentiated cell. And then it can differentiate into tissue, plant organ, embryo, or even a new plantlet. And that plantlet, from the test tube, we can move to the real field. So some terminologies are important here. In our right hand side, you can see the plant, a plant from root, shoot, apical bud, leaf, or apex. You can cut small part and grow in this test tube containing the media. So the small part, living tissue, transferred from a plant cell to an artificial medium for culture. This small part is called explant. It can be any portion of the shoot, leaves, roots, flower, or cell from a plant. Okay? Explant. Now you understood what is explant. Explant means the part of the plant which we uh, get in on the culture medium to grow and get new plants. So explant in tissue culture, very important. Other terminology is in vitro culture. I already mentioned you that in vitro means within the glass. It is a Latin word performing an experiment in a test tube. So tissue culture is done in the closed container, test tube, or even petri dish or glass bottle. So all types of culture, including animal cells, in vitro fertilization, etc. So in vitro means especially animal in vitro fertilization is also done, sperm and ovum fertilization, which is called in the uh, in vitro culture condition. Tissue culture, inclusive term for the growth of cells and tissues in a sterile environment. This is very important. Aseptic environment or sterile environment. What is it? That means in this condition, only the your targeted cells you allow to grow without any interference from other organisms. So undifferentiated plant cell you take, then plant callus or plant tissue, they then grow and differentiate into the organ. Another very important term, very closely related to tissue culture, is called micropropagation. Micropropagation is one kind of propagation. Tissue culture based propagation, the production of whole plants from a small section of a plant called explant. That is, from an explant, if you grow a whole plant, the process, and grow it in the field, it is called micropropagation. Explant can be apical bud, uh, axillary bud, or meristem. Meristem means dividing cells. In any eukaryotic organisms, all cells are not dividing at the same time. So, for example, tip, root tip, or shoot tip, or you know, a bud, they contains meristematic tissue, meristem. So you can target the meristem high potential to divide on the nutrient media. Usually, the method used by commercial tissue culture in the laboratories is micropropagation. What we can expand the definition of micropropagation. Micropropagation is commercial propagation of the plant uh, using tissue culture in the laboratories. Since because we can do propagation in many ways, stem cutting or grafting or seed, mostly seed is used for propagating unit. But if we use tissue or cell to make the whole plant seedling and grow into the plant commercially, then it is called micropropagation. Since a whole plant, including shoots and roots, is produced, which is genetically identical to the mother plant. This is very interesting. Micropropagation and other propagation, there are some difference. The most important is you use the tissue or cell of a plant in aseptic condition in the test tube in the laboratory and just make copy using nutrient medium. And in that case, mostly the seedlings are developed through the tissue culture process. They are almost identical, like the mother plant, that is explant you took. So this is an advantage. You can get homogeneously all the plants containing same genetic materials. But in case of seed and seed, when you use as a propagating unit, then there is a chance, because if the plant is a cross-pollinated, then there is a chance of genetic contamination. So micropropagation is very nice. Another advantage I shall tell you later on. In case of micropropagation, the seedlings we are using in agriculture, they are free from virus and other microorganisms. So what are the importance of plant tissue culture? Uh, the most important contribution made through plant tissue culture is the demonstration of the unique capacity of plant cells to regenerate full plants via organogenesis or embryogenesis. That is organ development or embryo development, irrespective of their source, root, shoot, uh, stem, floral parts, pollen, endosperm. That means any uh, vegetative or reproductive cell you can culture, and then by using different hormone combination of hormones and other environmental condition, you can trigger the culture cell into transform into the whole plant. And even you can produce the Haploid, diploid, triploid, because in tissue culture, if you take a pollen um, from the male organ from a flower, then it would be haploid. And you, by treating the haploid with colchicine as some agent, chemical agent, you can make diploid. So even you can fuse the cell uh, and make the diploid, triploid, or like that. So plant tissue culture is the best technique to exploit the cellular totipotency of plant cells for numerous practical applications and offers technologies for crop improvement. This is very important. Crop improvement, that is improving the traits of a plant, for example, making haploid, triploid, or in vitro fertilization, hybrid embryo rescue, variant selection, clonal propagation, or micropropagation, or even genetic engineering. Plant tissue culture is necessary because engineering or genome editing we do at the cell level. And if you change the DNA in the cell and then you need to culture it and get the new seedling or plant, and that is genetically edited or modified. So we cannot actually edit or engineer a standing plant. Uh, Another point is virus elimination. In most cases, for example, uh, potato. Potato is a famous and economically important crop in Bangladesh and worldwide. Potato usually we propagate by using the stem, the one uh, uh, we eat. So there are some eyes that germinate and make the small plant, and that you cut the potato and put into the soil. So yeah, lots of our food is used for the cultivation or uh, but nowadays you can produce potato in the tissue culture potato seedlings in Bangladesh. Bangladesh is champion. The bumper production of potato in last decades is due to the introduction of tissue culture technology for uh, micropropagation of potato. And micropropagation allowed us to get virus-free seedling, but traditional uh, potato tuber used for as seed, they contain virus. This is why the genetic regeneration or the uh, yield potential dramatically decrease. But using tissue culture, virus-free seedling give the highest yield. So Bangladesh, if you are asked a question, which crop has been commercially uh, cultivated in Bangladesh using the tissue culture technology. The number one should be economically important and robustly used potato. And the second, the orchid, banana, or some other ornamental plants commercially being used in tissue culture. Another thing is germplasm conservation. Some of the plants, they are um, uh, in the verge of extinction. So rare plant. In that case, you can get the tissue and make the copies and even conserve the plant. And some plants, they cannot produce seed. In that case, you can use the tissue culture. So production of industrial phytochemicals, another point. Phytochemicals, we have lots of medicinal plants. They produce secondary metabolites useful as phytochemicals for our medicine, cosmetics, and others. In that case, if you cut the plant and extract the phytochemicals, uh, it is not wise because you need lots, cut the plant and destroy the plant. It is uh, against the environmental balance. In that case, if you do the tissue culture and produce the phytochemicals in big fermenter, then no need to cut the plant and grow the plant and require the space. So this is very interesting. Uh, so tissue culture as a cell culture industry, you can culture the cell in big 5,000 or 10,000 liters, fermenter, offer the nutrient and trigger their cell division, and then extract the phytochemicals for your own industrial interest. Other point is regeneration of plants from genetically manipulated cells or by recombinant DNA technology or cell fusion, somatic hybridization. Two cell can be fused, and DNA recombinant DNA technology in earlier class I discussed. It is done at the cell level, and even which we call genetic engineering, even gene editing we do at the cell level. So in all cases, for crop improvement, advanced biotechnology, we need tissue culture. And tissue culture is a prerequisite in most cases. Plant tissue culture has extensively been employed for basic studies related to plant physiology, photosynthesis, nutrition of plant cells, and embryogenesis or embryo development, biochemistry, cellular metabolism, morphogenesis, organogenesis or embryogenesis, phytopathology, plant pathology, that is plant microbe interactions, even not only plant pathology, general interaction of plant cell with the microbe, then histolog, cytodifferentiation, cytology, cell cycle. Uh, Lots of basic research are being carried out using the tissue culture because uh, at cell level or tissue level, how the changes occurred, easy to study than the complex whole plant. Indeed, the discovery of first cytokinin is based on plant tissue culture. So I used one coined a term cytokinin. Cytokinin is a phytohormone. Plant tissue culture, um, many phytohormones are used. Cytokinin is one of them. So cytokinin is one of the most important plant hormone. So let me briefly share the history of tissue culture. History of tissue culture is very long. Plant, lots of people, thousands of people work on it. Among them, I shall name some of them. Uh, Dr. Gottlieb Haberland, a German botanist, made the first attempts to culture fully differentiated single cell isolated from the leaves of Lemium purpureum, petioles of Eichhornia crassips, and glandular hairs of Pulmonaria mollissima, and stamen hairs of uh, Trades Kensia in a simple nutrient solution of NOF. So he developed a nutrient solution and he found that these plant leaves and petioles, glandular hairs or stamen hairs can grow outside the plant. And the purpose of that experiment was to achieve division in these cells and obtain complete plants from them to verify the concept of cellular totipotency inherent in famous cell theory put forward by other researchers. Okay, so to prove that is Sladen and Schwann, these two scientists in mid 19th century, they gave the cell theory that is plant cell can be cultured and get the whole plant, that is it, they are totipotent, but To prove it, Gottlieb uh, did series of experiments and he was successful. And then Roger did some other experiment, the discovery of oxygen. He discovered and also recognition of the importance of B vitamins in plant growth gave the required impetus for further progress in the field of plant tissue culture. That means another hormone, a very famous hormone of oxygen and vitamin B vitamins are also important for plant tissue culture. Using indole acetic acid, this is indole acetic acid is an oxygen and B vitamins. Gautrat 1939 obtained continuously growing cultures from carrot root cambium tissues. And then we can name some more. Pierre, in the same year, White and Novecourt reported the establishment of callus cultures from tumor tissues of hybrid Nicotiana uh, glauca and Nicotiana Langsdorffi and carrot, respectively. These three scientists are credited for laying the foundation for further work in the field of plant tissue culture. These two guys, you must remember, and we must remember with highest honor. One is Toshio Murashige, another is Folke Skoog. And these two guys, they in 1950s, during 1950s, they at the University of Wisconsin, USA, made several major contributions. And most important is uh, Murashige and Skoog, they developed a culture media for plants tissue culture. It is still commercially available, called MS medium, Murashige and Skoog medium. Very, very famous. So Toshio Murashige and Folke Skoog, they popularize the micropropagation because combining different hormones and nutrients, minerals, they prepared a commercial medium called MS medium. So remember who is the discoverer of MS medium. MS medium still available and widely used for plant tissue culture. Which are the requirements for in vitro culture of plant? Most important, it must be done in the sterile environment that is free from the contamination. Define tissue culture medium. For example, Murashige and Skoog medium. Ex-plant tissue is very critical. Which plant tissue you are choosing for cultivation? Methods for in vitro propagation. What does it mean? Does it mean? Methods for in vitro propagation means all plants are not cultivable using the same protocol. Even within the same species, you may need to modify the conditions, even nutrient and other medium for getting a good result. So sterile work actually how we make things sterile because you collect the X plant from the naturally grown plant and then you must clean it and make it free from the microorganisms. So transfer plants to a greenhouse to reduce endemic contaminants, force outgrowth of auxiliary buds, remove surface contaminants by rinsing with sterilizing solution. So when you get the auxiliary bud or any other explant, there are some chemical solutions that can kill the microorganisms around the plant cells or tissues. Use detergent before sterilizing the tissue. So bacteria, fungi, mostly they are the contaminants. Unless removed completely, will overgrow the explants on the tissue culture media because tissue culture media, they are rich nutrient medium. If there are bacteria and fungi, they overgrow, faster grow than the plant cell. This is why you need to kill them and make uh, explant free from the bacteria, fungi, and other microorganisms. What are the sterilants we use to sterilize the explant to kill the contaminants? And they are mostly oxidants, different kind of oxidants, active halogens, heavy metal poisoning, powerful chemicals such as concentrated sulfuric acid may be used on seed. As far as possible, cut surface should be protected. There is always a trade-off between killing surface contaminants and killing the explants, because when you kill the surface contaminants, that is microorganisms, sometimes plant cell also be killed. This is why the plant cell has, strong cell wall composed of cellulose and hemicellulose. uh, Compared to other microorganisms, they are more tolerant. This is why maintaining a certain dose of the sterilants and a certain time, you can treat and immediately wash out the chemicals, allow the plant cells to grow on the medium. So here are some sterilants. Sodium hypochlorite concentration usually used 10 to 20%, and 10 to 20 minutes. So it is powerful halogen or oxidant. Calcium hypochlorite also being used, but sodium hypochlorite very popular. Hydrogen peroxide also people use. Heavy metal, mercury chloride or silver nitrate. Uh, they are also useful, but usually heavy metal is avoided. Antibiotics are rarely used because antibiotics, they have the selectivity, and some microorganisms have the resistance. Since many are merely bacteriostatic, massive outgrowth of cultures can result from residual bacteria, and or some bacteria, if resistant, even few bacteria or fungi, they can grow. This is why antibiotics are not uh, so powerful, rather than sodium hypochlorite and others are good sterilants. Here is the how we maintain the sterile condition or aseptic condition. It is a laminar airflow cabinet. Inside, you can do the job, and the air coming out from the hood, and not allow any microorganism, environmental microorganism inside the bench, so you can do the job in the clean bench. So surface sterilizing agents, there are huge. As I mentioned, you can get here depending on the what you are doing, in which plants are you doing. You use the suitable surface sterilizing agent. So schematic presentation of the protocol for surface sterilization, how step by step, here it is. First, you collect the explant, and then rinsing with water for 15 minutes, treatment with babystin for 20 minutes, another, sterilizing agent, rinse with water for 15 minutes, treatment with a TPOL for 15 minutes. That means step by step, wash with sterile deionized water, treatment with 0.1% mercury chloride for 10 to 20 minutes, sterile deionized water. These are the, this is only a generic one, but you can use other sterilizing agent also. Here you can see the agents used for surface sterilization, and how we, this is a tissue culture chamber, growth chamber, free from the micro organisms, so and under control light and temperature, humidity. So surface sterilization, sterilized explant cultured on suitable medium containing oxygen, cytokinin, a certain ratio, and redetermination, somatic embryogenesis taken place, and gradually you see first callus formation, and then gradually, plantlets developed. That is organogenesis taken place. First time the cell culture profusely dividing cell called callas, undifferentiated. They are uh, finally using different treatment. You can get the seedling um, and differentiate it to the plant. So tissue culture for disease-free seedling is very, very important. In case of potato, as I mentioned, stem tuber we use, ornamentals, color morph mutants, strawberry meristem culture. In our country, strawberry mostly the culture using the tissue culture, virus-free or fungal pathogen-free seedlings. Rooting of cuttings, conifer, grafting, palm, and many others. Banana, our country, also doing tissue culture. So many plants worldwide being commercially, especially ornamental plants, flowers, they are exactly maintaining the like mother plant tissue culture is being used. Reasons for in vitro cultivation of plant material, control environmental condition. Here I told you micropropagation is one of the important aspect. Other is pathogen-free materials and also phytochemical industry. Cryopreservation, you can preserve the tissue for the extinct, potentially extinct plant, genetic engineering, and also basic research. So in vitro pollination even you can do because you can do somatic hybridization. So these are some more advanced terminology, calogenesis, that is callus formation, embryogenesis, embryo formation, organogenesis, that is shoot and root development, rhizogenesis means root formation. So tissue culture can be different types. For example, meristem culture, that is you take meristem tissue and then culture. Another is embryogenesis from somatic cell, organogenesis from callus or directly explant. Adventitious shoot formation. Adventitious shoot formation is the de novo development of shoot from cell clusters in the absence of pre-existing meristem. That means different types of tissue culture is done. Factors determining the success of tissue culture, this is very important. Status of donor plant, that is which genotype, which species, age of the plant tissue, explant size, type, they are the determinant of the success of the tissue culture. Experimental condition, temperature, light, and day and night light, how many hours in the growth chamber you are maintaining. Composition of the culture medium, this is very, very crucial because composition is very complex and you need to find the appropriate ratio of different chemical constituents you are using in the media. So all factors are genotype dependent and require optimization for each cultivar. For example, tissue culture of rice, many our variety, they are easy, but some of them are recalcitrant in tissue culture. So plant tissue culture media composed of inorganic compounds, mineral nutrients. Actually, plant requires 16, at least 16 to 18 essential nutrient elements. What are those? Nitrogen, phosphorus, potassium, carbon, sulfur, manganese, iron, like in total 16 essential or 17 or 18. So you need to offer them in different ratios. Some are macro, needs more amount, some are very minute amount but essential. You need to add sucrose, carbohydrate, and plant growth regulators, that is hormones. Hormones like auxins, gibberellins, cytokinins, uh, abscisic acids. These are the hormones used in tissue culture, mostly auxin, gibberellin, cytokinins, and miscellaneous compounds, vitamins, and others. So this is really an art of doing the science. This is why you have to find out through the research which plant requires which combination of the nutrients appropriate for the in the culture media. So inorganic compounds here, I told you before, NPK, magnesium, calcium, sulfur, they are macronutrients. So need a larger quantity compared to the micronutrients, manganese, iodine, copper, cobalt, boron, molybdenum, iron, zinc, nickel or aluminum. Here are Murashige and Scoop, universal tissue culture medium. Here you can see they used macroelements, microelements, vitamins, and glycine, sucrose, growth regulators. They made a combination of minerals, vitamins, and hormones so that most of the plants can grow. But Murashige scoop is called universal tissue culture medium. Not sure your plant can grow exactly using that medium, but it is widely being used. Here you can see different elements are used as salt, so you need to calculate, and here they calculate. So measure growth regulators, which we call phytohormones, used in plant tissue culture are auxins, cytokinins, gibberellic acid, I mentioned before. What auxin does, it induces callus formation, root formation, cytokinin shoot formation, and embryogenesis, and gibberellic acid cell growth and cell elongation. So the combination of these three hormones are very crucial, especially auxin cytokinin. If cytokinin is the ratio is higher, then root never form, but a plant shoot will be formed. Here is the structure of chemical structure of auxin, like indole acetic acid, commercially available in the market, naphthalate acetic acid. These are the auxin. Auxin is a group of growth regulator. All these are auxins. So commercially available in the market, 2, 4D or NAA, IAA, they are available in the market. Auxin actually transport of auxin is basipetal. That is, here you can see it can uh, vertically go from top to bottom, never bottom to top. Very interesting in the plant. Lots of experiment being done. Um, Apical dominance, axillary bud dominance. This is due to the auxin formation. So auxins are very, very sensitive to photoperiodism, phototropism, apical dominance, cell division, differentiation, initiation of embryos. So another group I told you, growth regulators, cytokinins. In the market, you can find as kinetin or BAP, benzyl amino purine, or non-purine cytokinins also available. Cytokinin's main effect in tissue culture is adventitious shoot formation at higher concentration, inhibition of root formation, cell division, callus formation and growth, stimulus and outgrowth of auxiliary buds, inhibition of leaf senescence. So this is very interesting. Auxin and cytokinin act synergistically. Here you can see explant you put into on the medium. Here callus is formed. The ratio is, you can see auxin and cytokinin 3.3 milligram per liter, here 0.2 milligram. So if you are changing, here you can see root first form due to decrease of the kinetin. But if you increase the kinetin, shoot is formed. And if you decrease the auxin. So you need to have a balance. Even if there is no indole acetic acid or auxin, only kinetin and no growth. So you need to have a balance. So my effect of cytokinin and auxin on senescence. Senescence means, aging. So the plant leaves can be senesced, that is chlorophyll, dechlorophyllitis, or leaf green color damaged if auxin and kinetin that is oxygen and cytokinin ratio you used in the media. Here is the effect of oxygen and cytokinin. If oxygen is decreasing and cytokinin is here uh, increasing, then the ratio what will be developed, discussed here. For example, high oxygen root formation on the cuttings, low cytokinin. But if there is a balance, a callus initiation in dicot plants, if there is a balance between these two, you have to have the appropriate ratio. Gibberellic acid is synthesized from mevalonate pathway in the plant cell. So gibberellic acid is also very, very important, and it is involved in cell elongation. This is why in fruit orchard, people use spray a small quantity of low concentration of gibberellic acid to get the larger fruits. For example, the grape, vine industry they use, or even cucumber and many others. So this gibberellic acid can help the elongation of the cell. Here, an example you see no gibberellic acid dwarfing, but higher gibberellic acid elongated plant. So liquid media we used to support the protoplast culture. Protoplast, have you heard about protoplast? Protoplast is when a plant cell wall is removed and by different enzyme and other ways, then the membrane bound cell is called protoplast. We can also do suspension culture, homogeneous distribution by different ways. So liquid media also used for cell culture. Gelling agent, mostly in the nutrient medium, gelling agent is used agar. Agar, unbranched polysaccharide. It is red algae, for example, gelidium. Even in our country, some of the seaweed, they can produce the agar. Agar is very interesting. It can melt 100 degrees Celsius, and if you cool down, solidified gel-like structure. So this gelling agent is used in tissue culture, and nutrient media are combined and then autoclaved. So gelling agent can be gilangam also, another gelling exopolysaccharide from the pseudomonas. So hybrid propagation we can do by using uh, even hybridization can be done by uh, fusion of the cell in the cell culture condition. Mutation also. Even during the tissue culture, you can use chemical and radiation, physical mutagen to get the genetic variation. So crop, how to say, improvement at cellular level we can do. And many chemical compounds are produced. I mentioned the phytochemicals industrially in the doing the cell culture. So lots of tissue culture worldwide being done, and mm, uh, this is all about tissue culture talk today, and now it is open for you to ask some question. Hello, please ask some
Yes.
question.
Mm,
Just unmute yourself and ask the question or make comment. You can discuss freely. Yeah,
Hello,
now, yes,
sir.
yes, please.
Sir, if you don't mind, the slide for oxygen, can you
You
show the slide for oxygen again? The last slide, like right before cytokinin.
okay? I can go there. Just tell me stop like that.
Okay.
This
Yes,
one?
this one. Yeah,
Yeah.
yeah. I just couldn't finish writing all the points, sir. Sorry.
I shall give you the, the slides as well as hopefully the video record so that you can recap. But if you have any question now, you can ask the question. Do anyone have any question? No question? How many of you went through my article on Hamilton O. Smith? I shared with you because in my next lecture, I shall discuss on restriction enzyme and recombinant DNA, as well as the genome sequencing, genetic manipulation. So all of the uh, big discoveries and commercial use of the uh, genetic engineering, uh, the foundation was laid by Hamilton Othniel Smith, who was died recently at the age of 94 years. He first discovered the restriction enzyme. We shall discuss in a lecture later on.
Sir, is the article available in English as well?
English. This
Yes,
is a
sir.
good question. Yes, I can share with you the English version. But nowadays, if you find any article in the newspaper, you can choose in which language you are wishing to read. AI is now very powerful. So
Yeah,
if you
that's for
have,
sure. I can use the AI, but I have gone through the article. There was no option to choose the English one. That's why I was
I
asking,
see.
sir.
Okay,
Yes,
uh, I
I
can
had the similar question as
share
well.
with you the, an English version. Oh yeah, an English version you can find in my blog Medium. So Medium, Medium, is a blog where I publish the English version. So please visit there. If you visit Medium and put my name to Fajrul Islam or Hamilton O. Smith, you can find the whole article. Thank you. Good question.
Thank you, sir.
Welcome. Next.
স্যার আমাদের প্রিভিয়াস ক্লাসের যে স্লাইডটা স্যার লেকচার স্লাইডটা করানো হয়েছিল স্যার ওইটাকে শেয়ার করা হবে গ্রুপে
Yes. Today I shall share, the earlier lecture slide as well as today's lecture slide. And I shared a book, in biotechnology with all of you. And it is a PDF version of a complete book. So you can use freely, but never publicly share anywhere. Okay, it is copyrighted by the Springer. So for academic purpose, I am ethically allowed to share with you for only academic purpose, but they have the business. So please do not share publicly. Any other point? No? Any comment or question? In total, now we are 23. So I shall share. I'm going out in last class. I stopped somewhere, so I shall share some of the slides, remaining slides from that one. Let me connect it. Is it visible now? Is
Yes,
this
sir.
slide okay? Visible. We stop possibly here. That is the you know, genome sequencing. Genome sequencing cost is dramatically decreased. Now, even you can sequence your DNA by paying a little more than $1,000. And in my laboratory, we have three sequencers: Oxford Nanopore, very small one, and there are two others, Illumina sequencers. So sequencing is becoming very, cheap, and lots of, uh, sequencing of different organisms have been published in the cover of Nature, Science, and all top journals. And genomics tells us about our ancestry, uh, not only us, even all living organisms. We can, as DNA is the black box of all the genetic code of an organism. So we can get the hidden story of our phylogeny by sequencing the DNA. And in case of other organisms, also same. So in Bangladesh, jute genome was sequenced by, for the first time, it was a sensational topic and hugely been discussed. That is, jute genome was sequenced by Dr. Maksudul Alam, a Bangladeshi-born scientist who was a professor of the University of Hawaii, Manua. He voluntarily came to Bangladesh, and jute, the part is the was known as golden fiber of Bangladesh. It is not golden at this moment, but to get back the heritage, he sequenced the two types of jute, Deshi jute and Tosha jute genome, and published paper in Nature Plant. It was a very, a hugely discussed topic, and Dr. Maksudul was rewarded or awarded for his great job by the highest civilian award, Independence Award. After the jute genome sequencing, unfortunately, within few years after the sequencing, even before publication of the paper, Dr. Maksudul, he was my good friend. He died, and after that, the benefit of the jute genome project was not realized. And in Bangladesh, another big job, that is genome sequencing, was hugely being discussed. That is year-round fruiting jackfruit, Artocarpus heterophyllus, our national fruit. My group, we sequenced the whole genome of a year-round jackfruit, because jackfruit is seasonal. But year-round jackfruit, one tree found in Ramgarh in Chittagong, which gives year-round fruiting, very high number of fruiting. Anytime you visit there, you can find 200 fruits or like that. It was stunning. So I thought, if we get the mystery or molecular switches and codes of its year-round nature, then we can commercialize the jackfruit, because Bangladesh is the second highest jackfruit fruit growing country after India, and it is our national fruit. And then many industry can be developed. So this paper was published in Frontiers, and here the tree you can see, very interesting, and its fruits. It was hugely been discussed. Even former prime minister, who is now in India, left our country due to a huge movement. She was known Katal Rani or like that, due to discussed a lot about jackfruit after our discovery in an official celebration program at our university. She joined online. But anyway, this is one of the interesting study, and my team is still working on making the biomarker of and how to expand the year-round fruiting jackfruit worldwide. Okay, the many other molecular, I possibly discussed all these things, especially in molecular biology. Uh, Two scientists got Nobel Prize for their discovery of RNA interference, gene silencing by double-stranded RNA. Interesting. They found RNA technologies. They found uh, a double-stranded RNA. RNA is single-stranded that can be utilized to silence, undesirable gene very precisely. So gene silencing RNA technology is very, very important. And these three guys received Nobel Prize for their discovery, their studies of the structure and function of the ribosome. Ada Yonath, she joined online during COVID-19 when we organized World DNA Day. She joined online. They did a fantastic job, Benkit Ramon and Thomas. Ribosome is the site for protein synthesis, but ribosome structure of ribosome, 3D structure they determine, which is very complex and help us to understand how protein synthesis occurs at the ribosome. Even identical twin, when you sequence, you will find a different DNA sequence, not 100% same. There are some other interesting, discovery that mature cell can be reprogrammed to become pluripotent. In earlier lecture, I told you the plants are how to say totipotent or pluripotent, but in case of animal cells, mature cells or adult cells, for example, your skin, you cannot culture in that medium and get the whole, human or like that. In case of animal, it is thought that it is impossible. But Sir John Gordon, he tried at Cambridge, and he found that frog skin cell can be, differentiated into the other types of cell. That means it can be reprogrammed and make them pluripotent. And as a result, even can be possible to get the tadpole, that is the new frog. But at that time, people didn't believe his research. After his PhD, he stopped his work. Later, Shinya Yamanaka, just 50 years later, he actually was successful to develop mature cell as pluripotent. He found that several genes are involved in, blocking it to differentiate you into other cells. If you remove that gene, then you can make them pluripotent. And Yamanaka is very famous for stem cell discovery. So stem cells are pluripotent cells, and in medical science, stem cell therapy, even in our country, it is now becoming popular. So Gordon and Yamanaka received the Nobel Prize. Next time I shall discuss more about their exciting story. And another very interesting discovery was in chemistry was the DNA repair mechanism. That is, DNA, people thought they are immune, but actually DNA can be assaulted by environment. And they found that how DNA itself can repair, and they are very important for gene editing and other engineering. Other guys received Nobel Prize, for example, James and Tasuku. In medicine, they discovered the genes involved in immunity breaks, because there are some genes that work as a brake to the immunity. As a result, immunity has certain limit. But if you remove the brake, then immunity can be boosted up, and people thought their idea could be utilized for, uh, you know, uh, cancer treatment and others. And most, recently, Emmanuel and Jennifer, they got the Nobel Prize for their very profound work, that is genome editing, the sharpest molecular scissor used for editing. And they considered that tools can be utilized for rewriting the biology. And over the years, last 10 years, they discovered the genome editing in 2012. And then, around a decade, lots of things developed. Gene knockout, sickle cell disease treatment is now legalized in USA. And lots of experiment being done, several crops already been developed. For example, in Japan, gamma aminobutyric acid containing tomato, or high muscle producing red sea bream or tiger pufferfish used in delicate food sushi. The muscle protein is increased by genome editing, CRISPR-Cas genome editing. And disease screening, base editing, prime editing, and multiplex editing is being developed. And even our neighboring country, India, they developed 2 rice varieties using CRISPR-Cas genome editing. In Bangladesh, we from the Bangladesh Academy of Science introduced a commission project. I am the national coordinator. Many scientists like Professor Jeba Islam Shiraz, Professor Suhail Abdullah from Jahangirnagar University, Professor Shahinul Islam from Russia University, and many others, even the industry is involved in developing climate smart rice varieties. Next 10 years, people expect that gene editing can revolutionize in medical science, in agriculture, even curing the genetic disorder associated disease. You can edit and correct the mutation, even cancer, or obviously in agriculture, livestock, and poultry fisheries, it will be used hugely, and food security and sustainable development can be done. I am moving a bit faster to show you, GMO, perhaps genetically modified organism. Already I discussed with you the insulin, how we can do it, and GMO is obviously different from the conventional plant, because in case of GMO, compatibility for crossing is no need. So gene from any other source can be utilized. So it is revolutionary. Even gene editing is revolutionary. You can do the editing job at the DNA level. So here, genetic recombination using compatible sexual reproduction in plant or other organisms, no need. Source of the, if knowledge is clear, which code is involved in which a trait, then you can modify. Even in our food, naturally, GMO, there are lots of GMO naturally exist. Even gene edited organisms are naturally exist. For example, if I tell you sweet potato worldwide, all are transgenic, naturally transgenic. Similarly, the aromatic rice, which has one gene, BADH2, mutated. This is why naturally mutated. This is why the aromatic compounds are biosynthesized, not in other developed rice varieties. So it is a natural genome edited plant. So we, based on those knowledge, we can do in our commercial high yielding variety, we can make them aromatic and premium rice can be produced. So in Bangladesh, obviously BT brinjal, it has been commercialized since 2014, and golden rice still in the under the consideration in the environmental ministry whether it could be released or not. But in that case, rice is free from the vitamin A. Vitamin A is heat stable. So from the maize, beta carotene gene is introduced in the genome of our rice variety. So rice, golden rice, it looks like golden, carries the vitamin A. If it is released, vitamin A deficiency would hugely be solved. People expect that. So DNA research in Bangladesh, lots of people are doing, and lots of works are going on. So these are two lectures I have given to you. So if you have any more question, you can ask. After that, I shall share with you these two slides. So it is your turn to ask some question and make this session more interactive. Are you still connected?
Yes, sir.
Excellent. So let me talk about your quiz exam. As I mentioned, 3 quizzes. At least I shall take the 3 quizzes from you, and best 2 will be considered. So what about if we have a quiz in the next Saturday?
On which lecture, sir? Firstly?
First and second lecture, today's lecture. These 2
নেক্সট
lectures.
ইয়ারে তো অনলাইনে ক্লাস, অনলাইনে কুইজ হবে তাহলে।
নেক্সট লেকচার কত তারিখ হচ্ছে?
স্যার মোস্ট প্রবাবলি 14 তারিখ স্যার।
14th still online? 14th also online?
Yes, sir.
Yes,
Oh.
sir.
Then online quiz is not good. Then maybe 21st would be your
Okay,
quiz
sir.
exam. Okay. Any other question?
Sir, 21st February ছুটি থাকার কথা না?
Government holiday.
Yes.
Government holiday.
In that case, is it possible to arrange a makeup class on other day in person, because all online is not good. And for quiz exam, online is not good. Now,
Sir,
please discuss
like
whether 20th of February. I am not sure. Friday.
শুক্রবার।
স্যার
Friday.
20 তারিখ ফ্রাইডেতে স্যার ক্লাস কি ব্র্যাকে নেওয়া যায় নাকি? আই এম নট শিওর অ্যাবাউট ইট।
আমিও জানি না। তোমরা একটু খোঁজ করে দেখবা যে আমরা কি একটা মেকআপ ক্লাস অ্যারেঞ্জ করতে পারি কিনা, তাহলে কুইজটা নেওয়া যাবে।
I think it's possible, sir. Probably Friday in the morning.
হ্যাঁ, এটা তোমরা একটু কথা বলবা। হ্যাঁ, এই বায়োটেকের যে ইফতেখার সাহেব চেয়ারম্যান আছে, তারা অ্যালাউ করলে আমাদের জন্য কিছুই না। আমরা মর্নিং এ জাস্ট 1 আওয়ারের সেশন হবে।
A lot of master classes actually happens in the morning, Friday morning. Makeup classes usually.
ওকে,
নর্মাল ডিপার্টমেন্টের হয়ে থাকে।
Yeah.
যদি হয়ে থাকে তোমাদের দিক থেকে এই যোগাযোগটা একটু করবা। আমি যেহেতু ক্যাম্পাসে যাচ্ছি না রেগুলারলি, ফলে খুব ভালো হবে তোমাদের 2 জন ভলান্টিয়ার হিসাবে ডিসকাস করে এটা সেট করে ফেললে। আমাদের দরকারটা কি? আমাদের দরকার হবে জাস্ট একটা রুম রেডি থাকা। ঠিক আছে?
Yes, sir.
আদারওয়াইজ এটা টু লেট হবে তোমাদের এই টার্ম। দেখবা যে লাস্টে গিয়ে এই কুইজ একসাথে অনেকগুলো নিতে হবে এবং এটা টেরিবল।
Yes, sir.
ঠিক আছে?
কুইজ কত মার্কসের হবে?
কুইজ 100 মার্কের হবে।
জি স্যার,
100 মার্কের হবে। কুইজ কোশ্চেন 100 মার্কের করি আমি সাধারণত।
স্যার,
অনেকে 100তে 100ই পাই।
স্যার
স্যার,
এমসিকিউ হয় নাকি রিটেন হয় স্যার?
কুইজ কোশ্চেনই হয়। আমি জানি না কোন স্টুডেন্ট কি এখানে অ্যাটেন্ড করছো যে আমার কোর্স আগে অ্যাটেন্ড করেছো?
জি স্যার।
জি স্যার।
তাহলে যারা আগে অ্যাটেন্ড করেছো তারা এই ফেসবুকের মধ্যে একটা পোস্ট দিবা যে কি ধরনের কুইজ ছিল। আমি নিজেও তোমাদেরকে আগে একটা এই ধারণা দিব, আউটলাইন দিব, ঠিক আছে? কিন্তু
স্যার
অনেক
আমার
লেকচারের পরে কুইজে গেলে তোমরা খুব টাফ অবস্থায় পড়বে।
জি স্যার, ঈদের পরে এমনিও টাইম খুবই কম, মিড আছে,
ইয়েস,
দেন হচ্ছে আবার
ইয়েস,
কোরবানির
ইয়েস,
আগে আমাদের ফাইনাল হয়ে যাবে, খুবই শর্ট
ইয়েস।
টাইম।
হুম। সেই জন্য কুইজ হলে দেখবা যে অনেক কিছু বার্ডেন তুমি ফ্রি হয়েছো অনেকগুলো কুইজ হয়ে গেলে। কাজেই আমরা 20 তারিখটাকে টার্গেট রাখি। আর যারা আমার লেকচার আগে অ্যাটেন্ড করেছো কোর্স, তারা তোমাদের এক্সপেরিয়েন্স শেয়ার করবা যে প্রশ্ন কি ধরনের হয়। হুম। এখানে মাল্টিপল চয়েস কোশ্চেন এবং শর্ট কোশ্চেন থাকবে, ইলাস্ট্রেশন বা এরকম খুব এই তোমার সাবজেক্টিভ টাইপ বড় কোশ্চেন থাকবে না। কুইজ মানে হচ্ছে কুইজ, কিন্তু তোমার 100 নাম্বার থাকবে। 100কে ওই যে তুমি কমিয়ে 10-এ আনতে পারবা, কোনো অসুবিধা নেই, ঠিক আছে?
স্যার
ফ্র্যাকশনটা,
লেকচার
হ্যাঁ বলো। লেকচার,
স্যার লেকচার স্লাইড গুলো কি আপলোড করে দেওয়া যায়?
ইয়েস, আজকেই দেওয়া হবে।
থ্যাংক ইউ স্যার।
আর কিন্তু তোমরা কোনো সাবজেক্ট কি আসলে লেকচার শুনেছো?
স্যার আমার একটা কোয়েশ্চেন আছে।
ইয়েস।
স্যার আমাদের লেকচার ওয়ানে অনেকগুলো সায়েন্টিস্ট এবং ইয়ার দেওয়া, তো আমাদের কি সবই একদম মানে মাথায় রাখতে হবে? কারণ মানে স্যার ভুলে যাওয়ার সম্ভাবনা আছে, তো এর জন্য।
সব সায়েন্টিস্টের নাম মুখস্থ করা খুব বড় রকমের তোমার নলেজের ইস্যু না, কিন্তু তুমি কিছু ব্রেক থ্রু আছে, মনে করো জেমস ওয়াটসন, ফ্রান্সিস ক্রিক, অথবা তোমার হ্যামিল্টন ও স্মিথ, যারা এই আমাদের এই সাবজেক্টের, বা মুরাশিগে অ্যান্ড স্কুগ, অথবা তোমার আহ, আরো আরো কিছু আছে যারা, যেমন জাম্পিং জিন কে ডিসকভার করেছে,
বারবারা
বারবারা ম্যাকক্লিনটক। তো এরকম
ম্যাকক্লিনটন।
এইরকম ধরনের যারা খুব ব্রেক থ্রু করেছে, তাদেরকে আমরা ভুলে গেলে হবে না। কারণ তাদের সাথে সায়েন্সটা একেবারেই মিশে আছে। তোমরা জানো কিনা যে আমরা যে কোভিড-19 এই মহামারীর সময় যে ভ্যাকসিন নিয়েছিলাম, এই ভ্যাকসিন আবিষ্কার করেছিল ক্যাটেলিনা কারিকো, এবং তাকে নোবেল পুরস্কার দেওয়া হয়েছে। কি স্ট্রাগল সাম স্টোরি, এবং এগুলো একটু কিছু ব্যক্তির স্টোরি নিজেরা মনে রাখতে হবে, এগুলো তোমাকে ইন্সপায়ার করবে আসলে লাইফ যে কত স্ট্রাগল সাম, এবং সামথিং বিগ করার জন্য, বিগ কাজ করার জন্য এখান থেকে ইন্সপিরেশনও পাওয়া। কাজেই সব সায়েন্টিস্ট মুখস্থ রাখার দরকার নেই, কিন্তু তুমি ফার্স্ট রিকম্বিনেন্ট ডিএনএ টেকনোলজি এইটা ডেভেলপ করার জন্য তোমার এই পলবার্গ বা এরকম বিগ যে ব্রেক থ্রু গুলো আসছে, বা তুমি আরেকজন বিজ্ঞানী স্যাঙ্গার, ফ্রেডরিক স্যাঙ্গার, সে তোমার এই যে জেনোম সিকোয়েন্সিং, ইভেন প্রোটিন সিকোয়েন্সিং, সে 2বার নোবেল পুরস্কার পেয়েছে। এইরকম কিছু বিজ্ঞানীর নাম আমাদের মনে রাখতে হবে। এবং এইটা আহ, শুধু তোমার এই সাবজেক্টের জন্য না, মানুষ হিসাবে পৃথিবীতে এরা এত রুবাস্ট ইম্প্যাক্টফুল কাজ করেছে। এবং তাদের নামের সাথে যখন স্টোরিটা পড়বো উইকিপিডিয়াতে, উইকিপিডিয়া না, আমি সাজেস্ট করব ইমানুয়েল শার্পেন্টিয়ার তোমার জেনিফার, এদের তোমরা পড়বা, nobelprize.org-তে যাবা, দেখবা যে তাদের নিজের লেখা অটো বায়োগ্রাফি আছে। ফ্রি টাইমে মোবাইলও বসে বসে পড়তে পারবা, খুবই ইজি এবং লুসিড, পড়তে ভালো লাগবে, তখন সায়েন্সটাও খুব ইজিলি গ্র্যাব করতে পারবা। কাজে সব নাম কন্টিনিউয়াসলি মুখস্থ করতে থাকবা, ইয়ার মুখস্থ করতে থাকবা, এটা আসলে কোনো আমার এই লার্নিং অবজেক্টিভ না।
না স্যার, নামটা তো নামটা আমার লাগে পারবো আমরা, কারণ নামটা নাই করলে তাহলে কি মানে যে জিনিসটা ডিসকভার করা হয়েছে ওটাই তো, ওটা তো ওদের নামের সাথেই আর কি একটু রিলেটেড আছে, বাট
ইয়েস,
ইয়ার
ইয়েস।
নিয়ে তো আর একটু একটু এত করেন না, ইয়ার গুলো মনে
ইয়ার
থাকবে না।
ইয়ার ইন্টারেস্টিং না, ইয়ার
হ্যাঁ, ওটা
ইয়ার
ওটা
মেমোরাইজ
এত
করা
করবে
দরকার
না।
নেই, ঠিক আছে?
থ্যাংক ইউ
আর
স্যার।
আর এটা ভালো প্রশ্ন করেছো।
স্যার মানে কোন ধরনের কোনো ফিগার আঁকতে হবে না তো, তাই না স্যার, কুইজে?
না, কুইজে ফিগার রাখতে হবে না, কিন্তু তোমাকে ফিগার দিয়ে দিল, এটাকে লেবেলিং করতে হবে।
জি স্যার, বুঝতে পেরেছি।
ওকে না,
এরকম
লেভেল
থাকতে পারে।
ফাইন।
লেভেলিং মনে করো যে কোনো একটা খুব ইম্পর্টেন্ট কিছু একটা দেওয়া হলো, যেটা একেবারে ফান্ডামেন্টাল, কিন্তু কোনো একটা লেভেলিং মিসিং আছে, ওই জায়গায় কি হবে তুমি এটা লিখে দিবা। এটা সাধারণত কুইজে এরকম থাকে, এই সামনের কুইজে থাকবে কিনা নট নেসেসারিলি। কি লার্নিং পয়েন্টস গুলোকে আমি টেস্ট করব, কি এরকম না যে তোমাকে যে কোনো একটা লাইন উঠিয়ে দিল এটার একটা শূন্যস্থান পূরণ করো। নো, ভেরি ইম্পর্টেন্ট এই লার্নিং কি পয়েন্টস গুলোর উপরেই তোমাকে টেস্ট করা হবে। তারপর কনসেপ্ট গুলো, মানে মনে করো যে তোমাকে প্রশ্ন করা হতে পারে যে বাংলাদেশে এই দুটো জিএম ক্রপ, জেনেটিক্যালি মডিফাইড ক্রপ কাল্টিভেট করার জন্য অনুমতি দেওয়া হয়েছে এবং কাল্টিভেশন আছে, এগুলো কি, দুটা নাম লিখো, বিটি ব্রিঞ্জাল এবং বিটি কটন। তারপরে তোমার যে জেনিফার এন্ড ইমানুয়েল কি জন্য নোবেল পুরস্কার পেয়েছিল 2020-তে, তারা ক্রিসপার-কাস জিনোম এডিটিং টেকনোলজি ডিসকভার করেছিল। এইরকম ধরনের থাকবে কি, খুব তোমার এরকম না যে খুবই আনকমন ইস্যু। ঠিক আছে, কোনো প্রশ্ন? আরো তুমি ইভেন 4 মিনিট প্রশ্ন করতে পারবা। তোমরা শুধুমাত্র প্রশ্ন করো পরীক্ষা সম্পর্কিত, কিন্তু জানার কৌতূহলে প্রশ্ন করা দরকার। তো তোমাদের যদি আর প্রশ্ন না থাকে তাহলে আমি এখানে শেষ করছি। সবাইকে অসংখ্য ধন্যবাদ, এবং নেক্সট ক্লাসও তাহলে আমরা অনলাইনে নিতে হচ্ছে না। আমি আগেই তোমাদেরকে স্লাইড দিয়ে দিব যে কোন ক্লাস হবে, যাতে তোমাদের
অ্যাটেন্ডেন্স নেবেন না?
অ্যাটেন্ডেন্স অটোমেটিক্যালি এআই এখানে তোমার অ্যাটেন্ডেন্স রেকর্ড করবে, কে কতক্ষণ থেকেছে। ঠিক আছে। আর কি, এটাও ভালো প্রশ্ন। সব তোমার হচ্ছে কিভাবে পরীক্ষার রেজাল্ট ভালো করবা, তাই না?
স্যার নেক্সট ক্লাসে স্লাইড গুলো একবার রিভিউ করার পরে আমাদের অনেকগুলো কোয়েশ্চেন হয়তো চলে আসবে।
এক্সেলেন্ট, এটা খুব ভালো হবে। এবং তুমি ইভেন এই যে আগের ক্লাসগুলো রিভিউ করে যখন তুমি কোশ্চেন পাবা, এই কোশ্চেন গুলো ইমিডিয়েটলি তোমার আমাদের ফেসবুক গ্রুপে শেয়ার করতে পারবা, অথবা লিখে রাখলে এই নেক্সট ক্লাসের পরে, তুমি যেহেতু অনলাইন, তুমি কাগজে কোশ্চেন লিখে রাখবা এবং প্রশ্ন করবা। প্রশ্ন করা এটা একটা লার্নিং এর বড় প্রসেস, এবং এটা দিয়ে কারো ইন্টেলেকচুয়াল এবিলিটিকে জাজ করা যায়। এবং তোমাদের আসলে শেখানো দরকার হাউ টু আস্ক এ কোশ্চেন রাদার দেন উই আর আস্কিং কোশ্চেন এন্ড ইউ আর আনসারিং। এক্সামিনেশন সিস্টেম চেঞ্জ করা দরকার। আমার ইউনিভার্সিটিতে আমি পিএইচডি লেভেলের স্টুডেন্টদেরকে তোমার মনে করো একটা কুইজ পরীক্ষা হয়, যে 3টা লেকচার হয়েছে, এর মধ্যে ওখানে বসে একটা কোশ্চেন পেপার তৈরি করা, যে লেকচার গুলোতে সে অ্যাটেন্ড করেছে। যখন একজন কোশ্চেন করতে পারে, কোশ্চেন করতে পারলে তুমি সাবজেক্ট না যাই না প্রশ্ন করতে পারবে না, ভালো কোশ্চেন করতে পারবে না। ঠিক আছে, কাজে প্রশ্ন করাটাও তোমাদের প্র্যাকটিস করতে হবে। আর এটা জীবনের জন্য খুবই খুবই গুরুত্বপূর্ণ, একটা ভালো প্রশ্ন করতে পারা যে কোনো এই সিচুয়েশনে। ঠিক আছে, ওকে, ভালো থেকো তোমরা।